C3 is the most abundant complement protein synthesized by liver. C3 and C3 convertase are common to all pathways of complement activation. Complement cascade may be activated by one of the three pathways. 1 classical pathway. 2 alternative pathway. 3 lectin pathway. It begins with spontaneous hydrolysis of C3. C3a is an anaphylotoxin and chemoattractant. C3b is very reactive and can bind to invader's cell surface. If C3b cannot find the cell surface to bind within 60 microseconds, it is hydrolyzed. Factor B binds C3b on cell surface. Factor D cleaves factor B, leaving BB, which has a chainsaw mechanism. C3BBB acts as C3 convertase which cleaves C3. This creates a continuous loop and many C3b can be deposited on the invader's cell surface. C3BBB together with another C3b molecule can act as C5 convertase which cleaves C5. C5b together with C6, 7, 8 and 9 forms MAC which makes hole in cell wall. Inactivated C3b acts as an opsonin. It is activated by antigen-antibody complexes. C1 is a complex made of hexamer C1q and serine proteases C1R and C1S. C1q binds to Fc region of antibody. Activated C1s cleaves C4 and C4b either binds to antigen-antibody complex or to the adjacent cell surface. C1s also cleaves C2 and the C4b C2a complex is the C3 convertase of classical pathway. C3b formed can either follow alternative pathway or bind to C4b C2a to form C5 convertase. C5b follows the MAC pathway. In the blood, MBL, mannose-binding lectin, binds to another protein called MASP, mannan-associated serine protease. When MBL grabs its target, mannose on the surface of a bacterium, for example, the MASP protein clips C4 and C2 and together functions like a convertase to clip C3 complement proteins to make C3b. Normal cells are protected from complement by 1 membrane cofactor protein, or CD46. 2 decay accelerating factor, DAF, which is deficient in paroxysmal nocturnal hemoglobinuria. 3 cell surface protein CD59, protectin.